Over 20 years ago, Trina McMahon got interested in a freshwater lake. She's a microbial ecologist at the University of Wisconsin-Madison, and the lake is Lake Mendota. And there were other researchers who had already been studying this lake for years.
They study the fish, they study the plants, they study everything else but the microbes. And so I come along and say, hey, I'd like to study the microbes. And they say, great, just tag along. And so we have access to all their infrastructure, all their historical data. We have archives of samples going back to 1999, even for the microbes.
So Trina started working with the JGI to sequence and analyze all of those samples. It was a 20-year collection, a ton of information.
So we collected 20 years worth of samples and kept them in the -80 freezer. And we'd done some analyses along the way, but when the sequencing technology got advanced enough, we decided that was, the perfect time to go back for the full 20 years and do the metagenomes.
So the way that we collect the samples is we filter water and then we save the filter. So the sample is the filter. So it's old and it's precious and it's irreplaceable, and you want to make sure that you don't make any mistakes.
The samples of the actual bacterial biomass, Robin re-extracted. Logistically, it was very challenging to find the samples in the freezer.
I went through 2 -80 freezers and looked at every tube in them, looked at all the old lab notebooks from the lab, and I know people's handwriting from 20 years ago, so that I could look at the freezer tubes and be like, this is this person's project and he was working on Mendota. This is probably part of the time series.
So she spent probably almost a year planning to do all of that and actually doing it.
1,000 samples was a lot. And I feel like when people are routinely doing these large projects, they often have robots do the extractions for them, but I just was the robot. So this is what it looks like when we're preparing the plates to send to JGI. So all of the extractions are in different little tubes, and then I take out a little aliquot from each tube and put it into each well of the plate. And then we send all the plates of samples to JGI. And when JGI gets them, then they put it into their robots that do all the robot pipetting.